Welcome to the deep dive. You know how sometimes you just think about stuff like what happens when cells decide to just die?
Yeah.
So
Uh,
that's what we're diving into today.
Oh,
Apoptosis, or what some people dramatically call cellular suicide.
right?
We found this awesome article from Assay Genie called Apoptosis Caspase Pathways, a closer look at cellular suicide, and it's got some wild facts in there.
It's pretty amazing how our bodies just have a built-in self-destruct sequence for cells.
Yeah, it's not like some chaotic explosion, right? It's like super organized.
Yeah, exactly. It's more like this carefully planned disappearing act that keeps our bodies working, right?
From shaping tissues when we're just tiny embryos to getting rid of cells that could hurt us.
Apoptosis really is the key.
Okay, so it's like controlled demolition then, instead of everything just falling apart.
Yeah, I like that analogy.
So what makes it such a big deal that these cells die in this organized way?
Well, think about it. If a cell just randomly pops, it could release all this junk into the surrounding tissue,
Uh-huh.
triggering inflammation and maybe even hurting nearby cells.
Oh, so it's like a chain reaction.
Yeah.
Okay, that makes sense.
Apoptosis makes sure everything stays clean and contained, so it minimizes collateral damage.
So it's not just about dying, it's about dying the right way.
Right.
But how does a cell even know when to start this whole self-destruct thing?
There are two main pathways that get apoptosis going: intrinsic and extrinsic.
Hmm.
The intrinsic pathway is activated by these internal distress signals. It's kind of like if a city was under attack and raised the alarm.
So the cell's freaking out, and then it's like abort mission.
Yeah, basically. Things like DNA damage, stress, or if the cell doesn't have what it needs can all set off that alarm system inside.
So the cell is like, it's too much, I'm out.
Yeah, and it decides it's better to sacrifice itself so everything else is okay.
Wow, noble cell.
Then there's the extrinsic pathway, which is triggered by signals from outside the cell.
Okay, so like what kinds of signals are we talking about?
It's kind of like if the cell got an eviction notice from the surrounding environment, you know? This usually involves special molecules called death ligands that attach to death receptors on the cell's surface.
Death ligands. Death receptors. It sounds intense.
It is, but it's all part of this intricate system.
So one pathway listens to the inside of the cell and the other listens to the outside.
Right.
But both lead to the same ending, apoptosis.
Yeah.
So who are these key players that actually carry out this cellular suicide mission?
The executioners, if you will.
Hit me with it.
They're called caspases.
The caspases, okay.
They're this family of proteins that act like tiny little scissors, cutting up the cell from the inside.
Caspases, they sound kind of scary.
Well, they are pretty efficient at what they do.
So how do they actually go about demolishing a cell?
It's a whole process with multiple steps,
Mm-hmm.
and each caspase has a very specific job.
Like a demolition crew.
Exactly. They activate each other one after the other like dominoes.
Wow.
And that leads to the breakdown of the whole cell.
It's not just a random meltdown then?
Nope, it's very organized.
So the caspases target the main parts of the cell, chop up its DNA, and then what?
Basically, the cell breaks apart into neat little packages called apoptotic bodies.
Apoptotic bodies, okay.
Yeah, and then these fragments are swallowed up and taken away by cleanup cells.
It's amazing how the body just takes care of everything.
I know, right? No trace left behind.
So if caspases are like these super powerful executioners, how come they don't just go rogue?
That's a great question.
It seems like there must be something to keep them from just triggering apoptosis randomly.
Oh, absolutely. There has to be some serious control in place.
Like checks and balances.
Exactly. So apoptosis regulation is super important.
Gotcha.
It's all about finding that balance between signals that want the cell to live and signals that want it to die.
Hmm.
And a big part of this decision-making comes from a family of proteins called the BCL2 family.
BCL2. Okay, I'm taking notes.
Some of these proteins are pro-apoptosis.
Meaning they want the cell to die?
Yep. And others are anti-apoptosis. They're like the bodyguards. So there's like this constant back and forth inside the cell with these BCL2 proteins deciding what's going to happen.
It's wild how much is going on at this tiny level to keep us alive and kicking.
It really is incredible.
But what happens if the system gets messed up? What are the consequences of apoptosis going wrong?
Well, that's when things can get pretty bad, and we see a whole range of diseases popping up.
Oh, no.
For example, if cells are supposed to die but they don't, you can get uncontrolled cell growth, which is what happens in cancer.
So those rogue cells we talk about, the ones that avoid apoptosis, they're the ones that can form tumors.
Exactly, they become immortal.
That's scary.
And on the flip side, if there's too much apoptosis, that can also cause problems.
Oh, how so?
It can lead to the loss of important cells, which is a big factor in neurodegenerative diseases like Alzheimer's and Parkinson's.
So in some cases, cells are too stubborn to die, and in others, they're too eager to go.
Exactly.
So finding that balance is really key.
It is, and that's why we need to understand how apoptosis works.
Because then maybe we can fix it when it goes wrong.
Exactly. And it also opens up a whole new world of possibilities for creating therapies that can either make apoptosis happen or stop it from happening, depending on what we need.
That's a perfect way to move into the next part of our deep dive. We're going to be exploring how scientists are using this knowledge of apoptosis to develop new treatments for all kinds of diseases.
Pretty amazing stuff.
So we've talked about how apoptosis needs to be balanced. It's super important for us to be healthy, but it can also cause problems if it goes haywire. Makes me wonder, can we actually, mess with this whole process? Could we use what we know about apoptosis to make new treatments for diseases?
That's the big question everyone's trying to figure out, and it's super exciting. Imagine if we could tweak this self-destruct button in cells, boost it up to get rid of the bad ones or turn it down to protect the good ones.
Like switch to control whether a cell lives or dies.
Exactly. The potential is huge.
Okay, but how do you even start to manipulate something so complicated?
Well, remember those caspases?
Yeah.
The executioners.
Yeah.
They work in this chain reaction, right?
Mm.
So if we can come up with drugs that either make specific caspases work better or block them completely, we could potentially shift the balance of apoptosis.
So for cancer, we'd want to make the caspases go crazy and force those bad cells to die.
Exactly. And scientists are already working on this.
No way.
Yeah, there are some experimental therapies that are trying to directly activate caspases in cancer cells. And there are others that try to stop cancer cells from avoiding apoptosis.
So we could outsmart those sneaky cancer cells and make them self-destruct?
That's the idea.
That's amazing. What about those brain diseases we talked about? How can we use apoptosis to protect neurons?
In those cases, we want to prevent too much apoptosis because it kills off brain cells too quickly.
Right.
So researchers are looking for drugs that can block specific caspases involved in killing neurons, or that can boost the activity of proteins that prevent apoptosis in the brain.
So it's like giving those brain cells a shield to protect them.
Exactly.
Are there any drugs out there that are already using this approach?
There are. Venetoclax is a good example. It's already used to treat certain types of leukemia.
Oh, wow. I've heard of that.
It works by blocking a protein called BCL2, which is a key regulator of apoptosis, remember?
Yeah.
By blocking BCL2, venetoclax basically releases the brakes on apoptosis, so cancer cells can start dying off.
That's amazing. So understanding how apoptosis works at this molecular level can lead to real treatments. Are there other ways to target apoptosis besides caspases or BCL2?
Yeah, definitely. Researchers are also looking at ways to manipulate the death receptor pathway.
The one that gets triggered from outside the cell.
Right. Some experimental therapies use special antibodies that attach to those death receptors on cancer cells.
So it's like a targeted attack to kill those bad cells.
Yeah, you could say that. But of course, messing with something as fundamental as apoptosis has its challenges and risks.
Yeah, I bet. What are some of the things scientists are worried about?
Well, one big issue is making sure that these drugs only target the cells we want them to. We don't want them triggering apoptosis in healthy cells.
Oh, right. That would be bad.
Imagine if a drug meant to kill cancer cells also started killing healthy immune cells.
Yikes. So safety is a big concern.
Absolutely. Scientists have to do a lot of testing in the lab and in animals before they can even try these drugs in people.
That makes sense.
And even then, they have to go through many stages of clinical trials to make sure they're safe and effective.
So it's a long and careful process.
It is. Another challenge is that apoptosis isn't just a simple on-off switch.
Oh, right. We talked about all those pathways.
It's more like a giant web of interactions, with feedback loops and different pathways talking to each other. And it could vary depending on the type of cell and what's happening around it.
Sounds complicated.
It is. To develop effective therapies, we need to understand all those tiny details.
So it's not as easy as just flipping a switch, more like conducting a whole orchestra of molecules. What are scientists doing to tackle these challenges?
They're using really cool technology, like CRISPR gene editing.
Oh, wow, I've heard of that.
They can actually change the genes involved in apoptosis to see how specific proteins work.
That's incredible.
They're also developing ways to watch apoptosis happen in real time with special imaging techniques.
So they can see the whole process unfold.
Yep, it's pretty amazing.
So it seems like there's a lot happening in this field. What are you most excited about for the future of apoptosis research?
I think personalized medicine is going to be huge. As we learn more about how people's genes and molecules affect their apoptosis, we can start creating treatments that are tailored to each individual.
So like custom-made treatments for everyone.
Exactly. Imagine being able to predict how someone's cells will react to a drug based on their genes.
That sounds like science fiction.
I know, right? But it's getting closer all the time.
We've covered a lot today, from the tiny details of how cells die to the huge potential for new treatments.
It's been a wild ride.
And I think it shows that even in death, there's so much complexity and order.
I agree. The fact that cells have this built-in way to control their own death is pretty amazing.
Yeah.
And it makes you wonder, what else is out there that we haven't discovered yet?
Definitely makes you think about the bigger picture. Yeah, it really does make you think, doesn't it? I mean, we've been talking about apoptosis in just one tiny cell, but what happens when we start messing with this on a larger scale? Like for the whole body?
That's when we get into some really interesting territory and some potentially tricky ethical questions.
Yeah, for sure. Like we talked about how stopping apoptosis could protect neurons in those brain diseases.
Uh-huh.
But could that also mean people could live way longer than they're supposed to? Like, would we be messing with the natural order of things?
Exactly.
Yeah.
And what about using those apoptosis treatments to target very specific groups of cells?
Mm.
I mean, could that end up affecting other tissues in ways we don't expect? Or even change how humans evolve?
Whoa, that's getting pretty deep.
It is, right?
Yeah.
It feels like every time we make a big discovery in science, we also need to think be ourselves.
Totally. I mean, I started this deep dive just wanting to know how cells die, and now I'm thinking about life and death on a whole other level.
That's the beauty of science, isn't it? You
Yeah.
start with one simple question, and it leads you to all these new discoveries that change how you understand the world.
Well said. So to our listener out there, I hope you enjoyed this journey into the world of apoptosis.
It's been a fascinating topic.
It really has. We went from the tiny inner workings of a single cell to the big implications for treating diseases and even what it means for the future of medicine.
Definitely a lot to ponder.
It is. But remember, the learning doesn't stop here. Keep asking questions, keep exploring, and who knows what other incredible things you'll discover out there.
That's what it's all about.
Thanks for joining us on the Deep Dive on the Deep Dive.